Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. 1. To help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2. To build a supportive community where we can exchange the most valuable experience-based research advice. 3. To share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Okay, let's unpack this. Imagine a time when editing human DNA, well, felt less like precise surgery, maybe more like throwing a dart in the dark. What if I told you that nearly 40 years ago, a group of really audacious scientists, they didn't just hit the dartboard, they aimed for and landed a bullseye, making the very first, surgical strike on the human genome, not just randomly inserting DNA, but precisely targeting it. Today, we're diving into a landmark 1985 Nature paper. It's titled Insertion of DNA Sequences into the Human Chromosomal β-globin Locus by Homologous Recombination. You've shared this fascinating piece and some analysis around it, and our mission really is to cut through maybe some of the dense language and reveal why this study was, and still is, so incredibly important, a foundational step, really.
Yeah, it's fascinating how this paper just completely shifted our understanding of what human cells were capable of. Before this, the prevailing belief, even among top geneticists, was that the sheer complexity of mammalian DNA, especially human DNA, well, it made targeted gene insertion seem practically impossible, almost like a microbe-only kind of party trick. The thinking was our cells just didn't have the right machinery for such precise cut-and-paste work. This paper didn't just tweak that idea, it shattered it.
Right. So before this paper, if you wanted to get a piece of foreign DNA into, say, a human cell, it was essentially just a game of chance. You'd put it in and it would just randomly integrate itself somewhere, anywhere in that vast genome, that randomness.
Yeah.
That was a massive problem, wasn't it?
Oh, absolutely. It was a monumental roadblock. I mean, think about it. If you're trying to study the function of a specific gene by changing it, or even more critically, maybe aiming to correct a disease-causing mutation like the ones in sickle cell anemia or thalassemia, you have to hit a specific target, this kind of hit-or-miss approach. It meant that both fundamental research and, frankly, any real hope for gene therapy were severely limited. Which raises the big question. Could we ever move from these random insertions to something more like surgical editing in human cells? And, to really appreciate what they did, we need to understand the mechanism they were trying to harness. Homologous recombination. Think of it as your cell's really smart find and replace function for DNA. If DNA breaks or needs to swap info, it looks for an identical or very similar homologous sequence nearby and uses that as a template for accurate repair or integration. The scientists knew this happened in simpler things like yeast for decades, but many doubted it worked efficiently or, usefully in complex human cells.
And they weren't just picking any old gene either. They were looking at the human beta-globin locus. That's a really clinically crucial area, right? Not trivial. A lot of skeptics thought precise targeting in our big, complex genome just wasn't feasible, let alone in a gene so central to human health.
Exactly. Their motivation was this powerful mix of pure scientific curiosity. How does human DNA repair work? And a very clear application-driven vision. Could they actually correct these severe blood disorders by fixing the gene right at its source? Success wouldn't just validate human cells as editable systems. It would bridge that huge conceptual gap between simple organisms and the challenge of human therapy.
Okay, so here's where it gets really interesting. The experimental design. Given all that skepticism, the randomness problem, how do they actually manage this precision? They didn't just have a bold idea. They had a seriously clever plan. Tell us about the how.
Right, they combined three really critical innovations. First was the sophisticated design of their genetic payload. They engineered a special plasmid. Now, a plasmid is usually just a small circle of DNA, but theirs. It was custom built as a precision tool. It contained a 4.3 kilobase segment of the human beta-globin gene, specifically designed with what we call homology arms. These are long stretches of DNA that exactly match the sequences on either side of the target site in the human genome. And this was a key insight from Smithies. The longer these arms, the better the chance the cell's own recombination machinery would recognize and use them.
Ah, okay. So they built this specific delivery vehicle with these long matching bits to guide it, almost like a molecular GPS, to the exact spot on the chromosome.
Precisely. And crucially, embedded right in this plasmid, they engineered an echorrhea site. That's a specific molecular landmark, like a cutting site they put there deliberately. This let them later snip the DNA at that exact spot, which was vital for proving the plasmid landed exactly where intended. And what's also fascinating here, the plasmid was rescuable. It carried an origin of replication. This rescuable feature was brilliant, like putting a unique address label and a return key on their genetic package. If it integrated correctly, they could literally rescue that specific piece of DNA out of the human cell, replicate it in bacteria, and then sequence it. That was the ultimate proof. Real, precise targeting, not just luck.
That is incredibly elegant. So how did they get this super engineered package into the human cells, and then how do they find the needle in the haystack, the successful hits?
Right, that takes us to delivery and selection. They introduced this engineered plasmid, along with another marker gene, this one for G418 resistance, and antibiotic resistance gene, into human lymphoblastoid cells using co-transfection.
Lymphoblastoid cells, why those specifically?
Good question. They chose those, a type of easily grown human blood cell line, because they were relevant to the blood disorders they ultimately hoped to treat. Makes sense, right?
Okay.
So after introducing the plasmids, they grew these cells in the presence of the antibiotic G418. Only cells that successfully took up and actually expressed the G418 resistance gene would survive. Now, the crucial part here, even if random integrations of their main plasmid dominated, which they expected, this clever selection method let them find the rare, targeted ones, it was kind of like putting a super magnet in a haystack that only attracted the specific needles they were looking for.
Wow. But finding those rare ones, even among the survivors, must have still been a monumental task. How did they confirm they'd actually hit the beta-globin gene target and not just, some random spot that happened to pick up the resistance marker too?
Yeah, that's where the validation and feedback part came in. And this required immense scientific rigor. Immense. From about 500 antibiotic-resistant cell colonies, remember, these survived but weren't necessarily targeted correctly. They had to screen very diligently. They used a technique called southern blotting. That's a molecular biology method to find specific DNA sequences in a big mix. They were looking for very specific new DNA fragments that would only appear if their plasmid had precisely recombined with the beta-globin locus exactly as planned. This was the key diagnostic step. And after weeks, literally weeks, of laborious, painstaking screens, they isolated just 2 clones.
Just 2 out of 500. Wow. For a moment, that might sound, low. How did they spin that into a major success?
Well, just 2 might sound rare, but the real significance wasn't just the number, it was the frequency and the precision. They calculated a targeting frequency of about 4 times 10 to 3 per G418 resistant colony. Now that number, that was orders of magnitude higher than many skeptics had predicted was even possible for human cells. And the ultimate undeniable proof. That came when they sequenced the DNA from those rescued cells. It confirmed the recombination had happened precisely at the intended spot, the exon 2 site within the beta-globin gene. No unexpected rearrangements, no off-target integrations. This showed, conclusively, that if you use long enough homologous DNA sequences and a smart drug selection strategy, human cells could be coaxed into accurate, targeted gene exchange. It wasn't a fluke. It was real, reproducible, even if rare back then.
This kind of groundbreaking work, it's never done in a vacuum, is it? It sounds like a true team effort, especially pushing through those weeks of tough screening. Who were the key players here? What made their collaboration work so well?
Absolutely. It was a multi-institution effort, a real blending of specialized expertise. You had Oliver Smithies' lab at the University of Wisconsin. They were already experts in hemoglobin genetics, understood homologous recombination in simpler systems, and provided that conceptual backbone. Then collaborators at Baylor College of Medicine contributed crucial human cell lines. And Raju Kucherlapati's group offered vital insights into the selection markers and, importantly, the tricky southern blot design. This fusion was just essential.
So a real melting pot of different specializations. Were there any standout individual contributions that really, made the difference?
Oh, definitely. Oliver Smithies, who, by the way, later won a Nobel Prize for his work in this field. He brought the truly daring hypothesis. His conviction was that the inefficiency people saw in mammals wasn't like an absolute barrier, wasn't impossible, just a quantitative one, a rare event you could overcome. His insistence on using those long homology arms in the plasmid, that proved totally prescient. Then there's the graduate student, Ronald Gregg. Incredible tenacity, can you imagine? Doing those laborious screens, week after week of negative results. The sheer grind. Sallie Boggs refined the crucial DNA extraction methods needed for clean southern blots. That's often overlooked, but it's a decisive technical step for reliable results. And Kucherlapati's strategic advice, especially on the rescuable plasmid origin that provided the definitive proof. Real recombination, not just random stuff. It was just a remarkable mix. Conceptual audacity, relentless bench stamina, and methodological precision.
Okay, so let's connect the dots. What does this all mean for us today? This paper, 1985, that's decades before we heard common terms like gene editing or CRISPR. How did this work really lay the groundwork for what's possible now?
Well, if we look at the bigger picture, this paper wasn't just another piece of data. It fundamentally shifted scientific theory. It confirmed, beyond any doubt, that human cells have this robust, homologous recombination machinery that scientists could actually engineer and direct. This completely overturned that old belief we talked about, shattered the idea that precise gene exchange was somehow just for microbes. It showed human cells had this innate ability for precise gene cut and paste that we could learn to harness.
So it proved human cells are way more editable than previously thought. That's a massive theoretical leap. What about the practical side, the technology?
Yeah, technologically, it provided the very first practical blueprint for human gene targeting, period. It directly led to, or presaged, the development of knockout mouse technology. That revolutionized how we study gene function in mammals, letting us precisely disable or tweak genes in a whole organism. And then, yes, decades later, it directly foreshadowed CRISPR-mediated homologous recombination, the precise gene editing tool we hear so much about now. You know, CRISPR provides the scissors to make the DNA break, but it's also in homologous recombination, the mechanism they proved was usable, that provides the patch or the new insert for truly precise editing and for applications. This paper gave a clear conceptual path toward correcting genetic mutations, like those in beta-globin for blood diseases. It directly influenced early gene therapy strategies, and honestly, it still motivates the entire clinical gene editing field today.
It really sounds like a truly foundational paper. Even with the technology being so early stage back then, it just opened a door many thought was locked shut.
Absolutely. I mean, yes, the targeting frequency was still low back then, required that intensive screening. And this specific approach was more about insertions than, say, precise single-letter corrections. But the advance was far more than just incremental. It really planted the flag for precise genome engineering in human cells. It provided the intellectual foundation, the methodological groundwork that made all the later breakthroughs possible. It essentially moved the field from just dreaming about gene editing to knowing it was fundamentally possible. Then, it started building better tools.
This deep dive into that 1985 Nature paper really reminds us, doesn't it? Even the most revolutionary breakthroughs often start with a daring question and a meticulous, sometimes incredibly tedious, experimental approach. What began as this seemingly improbable surgical strike on one human gene. It laid the essential groundwork for our ability to precisely edit the human genome today, leading us hopefully towards a future where some genetic diseases might become a thing of the past.
And that raises an important question for you, the listener, to maybe ponder. Considering how foundational this early work was for everything that followed in gene editing, what do you think is the next big conceptual or technological leap needed? What's missing to truly bring the full potential of genome editing into widespread therapeutic use?